Stem cells have the ability to transform into any other type of cell, which some animals use to regrow limbs. They have the potential to help us repair parts of the human body that have been damaged by injury or disease in medicine. However, bone has proven to be particularly difficult to regenerate. Bone originates from what is known as mesenchymal stem cells, MSCs, which are found primarily in the bone marrow. Collecting these cells is a painful therapy, and then it is very difficult to convert them into bone cells to a useful extent. Researchers from the Royal Melbourne Institute of Technology University have found a relatively simple way to make stem cells turn into bone cells quickly and efficiently using high-frequency sound waves. The team set up the experiment using a microchip that generates sound waves in the megahertz frequency band and directs them to stem cells put in silicone oil and placed on a culture plate. They found the optimum setting was to expose the cells to a 10 megahertz signal for 10 minutes a day and for a period of 5 days, which increases the level of conversion into bone cells. The researchers hope the technique, which has several advantages over procedures in use today, could be used to restore bone that has been lost due to cancer or other degenerative diseases. Amy Gelmi, a researcher at RMIT in Australia, said, "This method also doesn't require any special bone-inducing drugs and is very easy to apply to the stem cells." Today's experimental procedures to change adult stems into bone cells often use complex and expensive equipment and struggle with mass production, making widespread clinical application unrealistic. In addition, several clinical trials attempting to regrow bone have largely used stem cells extracted from a patient's bone marrow, a high painful procedure. This new approach is an improvement in all of those areas. Researchers have shown that it can work with many types of stem cells, including fat-derived stem cells, without causing pain when removed from the body. Chemical engineer Leslie Yeo from RMIT said, "We can use the sound waves to apply just the right amount of pressure in the right places to the stem cells to trigger the change process. The device is cheap and simple to use, so it could easily be upscaled for treating large number of cells simultaneously. That upscaling is the next stage in the process. Theoretically, this approach should work beyond a small lab test. Ultimately, it is the hope that bioreactors can be developed to process stem cells in this way. Once the stem cells have begun to differentiate into bone, they can be injected into the body at the site of injury or disease, or coated onto transplanted tissue ready to grow new bone." The team said the process eliminates the need for drugs that modify stem cells and makes the whole process much quicker and more efficient. The team plans to continue working on how to scale it up for real-world use.